Okay, so welcome to our presentation. We did our research on bioremediation of microplastics, kind of seeing if they can be like hungry hippos and eat all the plastic in the ocean, in the environment. Just a quick overview of what we're talking about. It's just why do we care? What is bioremediation and the efficacy of it, the results, and kind of what we concluded about what we can do.
So, what are microplastics and why do we care? Next slide. Microplastics are defined as particles of less than 5 millimeters or less in length. They have two categories, primary and secondary. The primary microplastics are like made at size, so like microbeads and cosmetics or industrial abrasives like sandpaper. Secondaries microplastics are like created from the breakdown of plastic products, like like a jug outside in the sun. They're usually broken down due to weathering, like erosion or photodegradation, which is like the sun beating down on it and like breaking it down over time. They've been found in the air, the land, the water, mostly the water because of runoff, like agricultural runoff would like wash it all back into the ocean or a lake or anywhere with water. And of course, they're made from plastic. Plastic are made of polymers, and because of the many kinds of polymers out there, there's a very large range of characteristics they can have. Like they can be soft, hard, stiff, elastic. So they're very cheap, they're very easy to make, and they're very favorable in many industries like hospitals for like single-use plastic disposable stuff like gloves or bedpans for sterility, but this still contributes to the issue. Next slide. An Australian study found that there's 14 million tons of microplastics on the floor, which is a pretty conservative estimate. Um, it's a lot. Next slide. Here you can see some examples of plastics on the floor. This is just what you can see with the naked eye, since 5 millimeters and less. It's a very small size. Next slide, please.
Yeah, so like Michelle says, there's a lot of microplastics on the ocean floor and just a lot of plastic in the ocean. At least 87% of marine life have ingested or encountered some sort of plastic in their lifetime. And also due to the high surface area to volume ratio of microplastics, it's really easy for contaminants to absorb onto the surface. These contaminants can be from runoff, from oil leakage, or just part of the plastic production process. And as an organism may eat microplastics, these contaminants enter their body. And as the organism moves through the food chain, these contaminants can undergo biomagnification, increasing the concentration of pollutants within an animal's fatty tissue. So from a high level perspective, basically microplastics are in the environment. They get eaten by organisms, and then eventually humans can consume them through sushi. This is mostly for microplastics in an aquatic system, but we do encounter microplastics on a daily basis, whether it be in our water bottles, our food containers, or exfoliants that we use.
So moving on to bioremediation, just to recap, bioremediation is the process in which microorganisms or any living thing is used in the process of breaking down or removing environmental contaminants. Next slide, please. So in our study, we found that the most common form of bioremediation was enzymatic degradation. We found that there were multiple types of bacteria and fungi that produce enzymes. So like Michelle said earlier, there are a lot of different types of plastic polymers, and these enzymes are specific to, in their function, to each plastic polymer. So there's not like a universal enzyme. How these enzymes work is that they catalyze reactions by reducing the energy required in the chemical reaction. For example, if you look at the figure below, there's a hydrolysis reaction and how that splits up polyurethane polymer chains. So what these enzymes do is they catalyze a hydrolytic cleavage in the chain, allowing water to react with the polymer. This then breaks the polymer into smaller monomers or smaller chains that can then be consumed as a source of energy or be oxidized. Next slide, please.
Okay, so the big question we aimed to answer through our research was whether microbial degradation is a viable and effective alternative for plastic waste remediation, especially common types of plastics such as like polyethylene, polystyrene, and polyvinyl chloride, better known as PVC. And so there are several studies that found mealworms to be a quite effective means of breaking down a plastic type called polystyrene, which we know as styrofoam. And so these mealworms were able to break down styrofoam at about 50% molecular weight reduction within a 24-hour period. And even better, the worms didn't seem to be negatively affected by usual toxicity levels seen in other microbes. Overall, through our research, we wanted to see, like, is it effective, right? And overall, we found that most plastic biodegradation usually requires a lot of different pre-treatment mechanisms in combination to actually produce effects. And even with this, microbes usually have to spend long periods of time ingesting plastics on the order of between a few days to even as long as a year before results actually show. And this is in contribution to what Matt was talking about earlier. Each specific plastic type has specific enzymes and fungi that like need to be used. And so you can't use one for every single type of plastic. And then, and so that's not as efficient. And the results that usually come up are usually pretty mediocre with about 10 to 30% molecular weight reduction on average. And most products of microbial degradation are only partially degraded polymers, which can also be much more mobile in soil and aquatic environments. And that can pose even more problems in the future. Next slide.
So with all remediation technologies, there are both positives and negatives. So let's start off with the pros. So for bioremediation, there's basically only two main pros. First is that it's not as invasive to the natural environment as some other forms of remediation. And second is that it's actually like a complete remediation process. It's like you have something harmful, right? Plastics, and then it actually gets broken down into something harmless. You get these really long chains and they're broken down into smaller chains, which are harmless for the environment because monomers are basically just food. Unfortunately for bioremediation is there's just so many limitations in trying to implement it both in site and out of site. So let's start on the left side here with the photos to see what's wrong with implementing it in site. So first and foremost is that microplastics are not a good source of food. Microorganisms just don't want to eat them. They're about as, like, that apple there is about as appetizing to a person as microplastics are to a microorganism. You would eat it if you're starving, but if there's literally anything else, you're probably going to eat that instead. So that's an issue because you have something you want to get cleaned up, you have a way of doing it, but they're just going to ignore it. And if you try to force them through genetic engineering to eat it, it's a really bad food source. So they're just going to get outcompeted. And in a few generations, you're not going to have any microorganisms left. Second, for doing an insight, you need a lot of pre-treatment, right? It's like the pizza in the oven. When you got to cook something before you give it to a person, right? And it's kind of the same thing with the microbes. You got to run it through some chemical, some physical, and some heat and radiation treatment before it could really efficiently get broken down. And lastly, for insight is that microplastics, because of their small size, are highly mobile. And ultimately, they end up in really large bodies of water, such as the ocean, where it's just too large. You can't do any pre-treatment and you can't do any control conditions at all because it's the ocean. It's just too big. Now let's look at what happens with limitations in terms of like doing it offsite. And really, the limitations here are going to be cost and time. The cost, because you need to find a way to gather all of the microplastics, which is a huge task in itself. That takes energy. Then you got to move it to a place where you can treat it. That also takes energy. Then you have to pre-treat it and you have to maintain the environment for the microbes. All of this costs money, right? And what you get out of it is just nothing you can sell. You can't just sell the monomers. You don't get any profit from using bioremediation. So there's no incentives to make like a startup to break down plastics, unless you're getting some kind of like government subsidy, basically. And lastly, for out of sight is it just takes a really long time. It could take months to years to just break down by a few percent. Next slide.
All right, so since we've determined that bioremediation isn't really that effective, the question turns to what can we do to limit the pollution? So the main thing you can do is to limit production, just stop it at the original source. That's kind of hard just because of how prevalent plastic is and how cheap and just how cheap it is. So another thing you can do is just collect it in different ways. So one way is like domestic ways is the Cora balls. It's basically you put it in the laundry and it can catch some microfibers. There's other similar technologies with this, and basically it catches the microfibers and prevents it from going out in wastewater. Another example is the sand filter. As you can see the people using down there, they filter the sand. It can catch pollutants as small as 100 micrometers across. And you can also like drag this behind a boat or something. But again, limitations, you need manpower, you need time, you need to cover a large area. So it's tough, it's doable, but it's tough. And then finally, there's this project that's going on right now in the EU. It's called Go Jelly. Basically, they're using the jellyfish mucus to help bind to nanoparticles and microplastics. And they're working on collecting pollutants that way. Next slide, please.
So in conclusion, the point of this project was to see if bioremediation was a good solution to widespread microplastic contamination in the natural environment. And basically we concluded that alone, like just using it by itself as a remediation technology, it's not the solution. But not only is bioremediation not the solution, but there is just not a single remediation technology that's going to take care of all the plastic contamination in the environment. There needs to be further research conducted into remediation technologies that can be used in collaboration together. There's no one solution that will fix all the plastic pollution in the environment. So really more research just needs to be done. And while we're trying to figure out a good solution, we just need to stop we need to stop putting so much plastic in the environment until we have an actual solution to it. And that's all.